Okay, good evening, everyone. Hopefully that's most people joined us. Any latecomers will let in. Welcome to our Cambridge Festival event entitled The Role of Genetics in Cardiovascular Health and Disease. I'll just give some housekeeping notices to start. Please ensure your microphone is kept muted through the presentation. Feel free to ask any questions in the chat at any time during the presentation, and we'll take these at the end when we have a Q&A, and we'll give you the option to raise your hand then as well. Another note is that we are recording this event, so if you don't want to appear on screen, please ensure your camera is turned off. So we'll begin by introducing who we are and where we're based. We'll then get on to the subject of cardiovascular disease and the underpinning genetics. We'll describe some of the studies we're carrying out, what we hope to find, and how the results will help improve early detection and treatment of heart and circulatory problems in the future. We'll also highlight the importance of this research and how you can get involved if you wish. So I'm Amy, and together with my co-presenters Valerie and Lisa, we will present work from our department to illustrate how we tackle the topic of genetics and cardiovascular disease. The principal investigators for this work, Dr. Poole and Dr. Harrison, will be available for a panel discussion at the end to answer any questions you might have. So if you do think of any as we go along, please do type them into the chat box. We are from the Cardiovascular Epidemiology Unit, which is based at Strangeways Research Laboratory, just across from the Addenbrooke's Hospital campus. We have close links with the hospital, and clinical measurements from our studies, which we'll get on to discussing later, are carried out in research facilities on the Addenbrooke's site. I'll just highlight that the MRC/BHF part in our name relates to the core funding we receive from the Medical Research Council and the British Heart Foundation. Our focus is on improving knowledge of causal risk factors for cardiovascular disease, preventative medicine development, public health interventions, population screening, and global health. We are part of a large department with many studies underway and many links with other research groups, for example, at the Sanger Institute and Health Data Research UK. So before we start, a quick poll to find out how much you, as the audience members, feel that you know about this topic, genetics and cardiovascular disease. So we'll give you a minute or two to answer the question. Okay, that's nearly everybody, just a few more left. Okay. So that's interesting. There's a good proportion of you with a little understanding. So hopefully our talk today will share some insights with you. Thank you very much, everybody, for participating. So to start off with some background, cardiovascular diseases encompass all diseases of the heart and blood vessels. They are the highest cause of death globally. In the UK, there are about 7.6 million people with cardiovascular disease. This equates to about 450 deaths every day, with more than 100,000 hospital admissions every year due to heart attacks. So this is a huge burden on our health service. There are many different types of cardiovascular disease. Focusing on circulatory disease, these can be broadly grouped into two categories. There are those that involve the narrowing of an artery. Examples include heart attacks, stroke, and peripheral arterial disease. Over time, the arteries can harden and plaque can build up, narrowing an artery and causing problems. The blood itself can cause cardiovascular problems by being sticky, predisposing to clots. The second category occurs when a blood vessel wall becomes weaker and the vessel bulges. This is known as an aneurysm and includes cerebral brain aneurysms and aortic aneurysms. So why do we get cardiovascular disease? The modifiable risk factors are that the risk of our genes is just one of a number of contributing factors. The modifiable risk factors include being overweight, diabetes, blood lipid levels, high blood pressure, and these are easily measurable. Socioeconomic factors can also play a part. Then there are non-modifiable risk factors, age, gender, and genetic variation. Some diseases are definitely hereditary, with conditions that are 100% genetic. In some, genetics plays a role, but may not be the sole component. For those, each cardiovascular disease has a heritability of 50 to 70%. The remaining percentage is environmental or other factors, as illustrated in the diagram here. Looking at the effects of genetic variation on the risk of cardiovascular disease, predisposition to cardiovascular disease is evident in some families and has been known for some time to be associated with mutations in the low-density lipoprotein receptor gene on chromosome 19. With the vast amount of information stored in our DNA, we need to find a way to home in on particular sites where variations can affect particular disease. In our case, we are interested in cardiovascular disease. This slide shows two versions of the same strand of DNA. Each is represented by an image and by the letters for the bases shown in the key. Adenosine always pairs with thymine and cytosine with guanine as they fit together in what is known as base pairs. According to the sequence, certain areas of the DNA, or genome, code for particular proteins. If there is a difference, i.e. variation in the sequence, it could affect the production and/or formation of the protein. Shown here are some differences between the two strands. It's not easy to spot them, and increasingly sensitive techniques have been developed to determine these sequences for study in the genomes of many individuals. So we have a quick question to ask how many differences you can see between the two strands of DNA and what you think the effect could be. So I'll just give you a second just to have a look and see if you can see any of these differences. So this next slide highlights in red the differences in the different versions of the code. You can see both the letters are different, the ones highlighted in red. A broad approach using large numbers of individuals has been to study a set of genetic variants genome-wide, that is, across the genetic material, i.e. DNA, in the different individuals to see if any particular variation is associated with a trait. These genome-wide association studies allow researchers to compare frequencies of people with and without disease. Because the risk of each of the variants on disease risk is quite small, large sample sizes are required, leading to the development of many large collaborative GWAS studies involving, in some cases, tens of thousands of cases and controls. In addition to the low-density lipoprotein association on chromosome 19 mentioned earlier, the first genome-wide association studies found a strong association for coronary artery disease with a locus unexpectedly on chromosome 9, and there have been a lot more discoveries since. So we're refining our search for needles in the haystack. Moving on from discovering where in the DNA there are variations that matter, we are interested in how these changes affect the construction of proteins, the pathways of intended tasks of the proteins, and how all the knock-on effects cause problems for the individual. When we fully understand these pathways, we can plan where to target treatments. Our research group in the Cardiovascular Epidemiology unit has been set up to try to answer those questions. Our mission is to identify and characterize causal biological pathways to find out exactly how newly discovered genetic variants, or genes, influence cardiovascular disease. To tackle this challenge, we combine a number of computational and experimental approaches to translate the genetic associations into causal biology. One such example is the so-called Recoil by genotype mechanistic studies in healthy volunteers. We will explore over the next few slides how these work and give some examples of our current studies. The volunteers in our studies are recruited from the NIHR BioResource. The BioResource is a national panel of over 200,000 volunteers with or without health conditions who are willing to be contacted to participate in research studies. These research studies investigate the links between genes, the environment, health, and disease. Upon enrolling in the BioResource, volunteers provide a blood sample and answer questionnaires on their medical history, demographics, and lifestyle factors. Individuals of interest can be identified from this database of genetic and/or physical, so phenotypic, features. And these can be then invited to take part in research studies. Enabling researchers to recruit volunteers for research studies based on their genetic makeup is a unique feature of the BioResource. The NIHR BioResource identify and approach volunteers who carry gene variations of interest on our behalf, and these volunteers can choose whether they take part in a study or not. Here we have an example of a Recoil by genotype study design. Healthy volunteers from the BioResource panel are invited to take part in the research study. This is the panel on the left. In the middle, you can see that participants are selected based on whether they carry a particular genetic variant, known as cases, or they don't carry the variant, and these are known as controls. The volunteers may be asked to undergo additional tests. Here we show some examples, including a dietary challenge on the right. Results are then compared between the groups to answer the research question. We have compiled a series of research studies to investigate the role of particular genetic variants in 3 areas. GemBio and CAD Bio are similar in that they are looking at genetic factors relating to cardiovascular diseases, but they differ in the complexity of the tests involved. CAD Bio is a new study we hope to launch soon relating to aortic aneurysms and dissection. On our behalf, the BioResource identifies individuals in their database carrying genetic variants, which have been established by previous studies to be of interest. So I'll now talk you through some of the studies. The GemBio study is the simplest of our studies. This is in terms of the interventional tests volunteers are required to undergo. Here, volunteers are asked to complete a questionnaire about their medical history, demographic, and lifestyle factors. We will take height, weight, and blood pressure measurements and ask them to donate one blood sample. We can measure a range of molecular and cellular biomarkers in the blood and also look at things like blood cell characteristics. The properties of these are then linked to the specific changes in the DNA that are known to associate with a person's risk of cardiovascular disease. We have 2 gene variants under investigation in this study. SWAP70 is a gene which plays a role in the immune response modulating cardiovascular disease risk. The recruitment of volunteers for this sub-study has not yet started. The GMPR gene has a role in blood cell formation. This sub-study is complete and was conducted by Lisa as part of her PhD. I will now hand over to Lisa to tell you more about her findings.
Thank you, Amy. Hello, everyone. Yes, so I'm going to talk you through some of the results of the GMPR study. And just to track back a little bit, the goal of these studies is to understand how genetic variants that we discover in these genome-wide association studies influence different traits that can be disease risks or also intermediate traits, such as blood cell-related things. And in the GMPR study, we were interested in a locus on chromosome 6. And basically what you see in this plot is the chromosome location, so the DNA at the bottom, and then the confidence of association. And each dot in this diagram is a genetic variant. And the height of the dot shows you how strongly associated it is with the trait of interest. In this case, we're looking at hemoglobin levels, hemoglobin being the molecule that carries oxygen around our bodies and red blood cells. And if you think about it, each variant can exist in the normal version. So you could have a C and you have one from your mother and one from your father. But then some people will have a different variation, so a different letter in the DNA, for example, a T. And what we see here is that an increase in hemoglobin levels is associated with having more letters T instead of the C. But then other variants in the same locus will not be associated, so they show up lower on this diagram. And so what we really want to do in these studies is kind of identify which of all of these variants actually have a biological function, which are actually changing biology in the cells, and which genes are affected, so that we can then develop new treatments and drugs to target these genes. And if you look at the bottom here, this is the genes that are present in this region, and these variants overlap with a gene called GMPR. So through a lot of computational analysis that I did in the beginning of my PhD, we identified 2 of these variants to be most likely to have a function. And basically there was one common variant that sits in an intron of the gene, so the part that's not forming the protein, but is just a regulatory region of the gene. And this variant is present in about a third of people, so in 36%. So about 1 in 3 of you might have this particular change in your DNA. And that variant was associated with having lower hemoglobin levels in red blood cells. And then the second variant sits in an exon, so part of what forms the protein coding region of the gene. And this one was a rare missense variant. So it's very rare, only present in 1 out of 500 people. And it increases the hemoglobin levels in red blood cells. If you think about what it means to be a regulatory or a missense variant, I put this little sentence at the bottom. So basically all the black parts are kind of words that form the message of this gene that is translated into a protein, which is kind of the molecular machines that float around the cells and do things like our metabolism and so on. And basically the normal message would be something like, the man saw the dog hit the cat, full stop. And then what a regulatory variant can do is change like how loudly you say the message or how much of that message gets out. So the regulatory variant might turn all those letters into smaller letters and you get less of the message. Whereas the missense variant that sits in this protein coding exon will change one of the letters. And then the challenge is if the sentence now reads, the man saw the sock hit the cat, we don't know what that means, right? We don't know if it's important that a dog hit the cat or if that's not that important. So really the challenge is out of all these genetic variants to identify what is the actual function, because we can't just translate that from the GWAS directly. So what we went ahead and did is conduct a Recoil by genotype study for these two variants. So we went to the NHR BioResource and selected individuals that carry different combinations of the rare and the common variant in that GMPR locus. And basically, I said you have one copy from your dad and one from your mom. And we didn't find people that have kind of two copies of the rare variant. So there were just no individuals in the BioResource. But we found 5 different combinations of having the rare and the common variant. So we recalled people in those categories. And in total, we managed to recruit 29 individuals. And these were all matched for age, sex, and body mass index, also things like smoking and other characteristics. And that's very important because that allows us to kind of eliminate factors that might influence how variable measures are between people. And so we can really focus on the genetic changes and compare people that are as similar as possible in other regards. And so those 29 people came into the BioResource in Cambridge and gave a blood sample. And from that blood sample, we measured different things, and they also filled out a questionnaire and had their height and blood pressure measured. So 2 things we measured were protein and metabolites levels, and I'm just going to talk you through how one of these assays works. So basically, we isolated red blood cells, so fresh red blood cells from the samples that we collected, because the variants are related to hemoglobin, which is present in red blood cells. So we could get kind of the cell type of interest. And then we processed all the samples for proteomic analysis using mass spectrometry, which is a very sensitive technique to analyze basically all of the thousands of proteins that we have in our cells. And once we looked at the protein levels for the GMPR gene, which is the gene that the variants sit in, we actually saw that if you split the protein abundance in the samples across the 5 different groups, we saw that people that had the rare variant had lower GMPR protein levels, and people with more copies of the common variant had higher levels. So actually, that is very interesting because it shows us that probably the rare variant decreases the levels of the protein. We had one outlier here, and so this person should not have had any rare mutation. It should have been the perfect control if without any of the variants. But when we looked back at the genotyping data from the BioResource, actually we found that this person had a rare Frameshift variant also in GMPR that was not previously known to us that it was associated as well. And a Frameshift is in a way much more severe than the missense, because it's like you delete a letter in the sentence and then the whole frame of words shifts. So you can't really read the sentence anymore at all. So given that this also lowers the protein levels, we are quite confident that the missense variant has a severe effect on the protein as well. So the effect between these groups was not statistically significant, so we grouped them all together as the control, and we grouped all the ones with a rare variant, the missense or the Frameshift, into this rare variant group. And for the next essays I'm going to show, we're kind of comparing this rare group of individuals to the control group. So let's think a little bit about what does GMPR actually do? What does this gene do? So GMPR is the GMP reductase, or guanosine monophosphate reductase. And basically it's an enzyme that is an important part of the nucleotide metabolism in our cells. So nucleotides are the basis of our DNA, so the little building blocks that make up DNA. And what you see here is kind of an outline of the biochemical pathway of how these nucleotides are created. And you see there's a lot of circular feedback going on. I put also some structures of the molecules that are produced on the sides. And basically, GMPR is the enzyme that catalyzes one step in this pathway from this guanosine monophosphate, GMP, to form inosine monophosphate, IMP. And we think, like people in the literature think this is important because it keeps the balance of this pathway between producing the G letter GTP and the A letter ATP. Also ATP is relevant for energy in the cell, and GTP and GDP for signaling in cells. So like this pathway is very central to a lot of cells. So if you have this rare variant in GMPR, we know from the protein data that the GMPR levels go down, but what happens to this pathway is the function actually affected. So we went and measured levels of all these metabolites, as we call them, or biochemicals, in the samples that we collected from the Recoil participants. And what we found is that basically the metabolites on the left side here that are kind of upstream of this GMPR reaction, when you have the rare variant, they increase in levels, which makes sense because this reaction doesn't happen as often anymore. So you accumulate levels of this and produce more GTP. At the same time, levels of the xanthosine monophosphate, which I put here, and xanthine were significantly decreased in those samples. So basically, if you don't get as much GMPR reaction, then these go down. Interestingly, this effect was observed in red blood cells that we analyzed, but not in blood plasma from the same people. So plasma is the liquid component of blood once you remove all the cells. And that is often studied in larger studies because it's quite easy to obtain that. But we only saw the effect in the red blood cells, which is where the variants likely act, because the hemoglobin obviously is only in red blood cells. So that again shows the importance of having access to the right cell type to observe these sorts of mechanisms. So the main result from this for us was that the rare variant, the missense variant, that is associated with hemoglobin levels, does affect the GMPR levels and also the function of this protein. And then we did proteome-wide analysis. So we have now a whole network of proteins that are differentially expressed between the people with the rare variant and the control group. So we can do things like network analysis to find out which pathways are affected. And we're also performing more mechanistic validation experiments in the lab to understand this mechanism and how it relates to hemoglobins. In general, the Recoil by genotype study design allowed us to really drill into this very rare variant, and at much greater power. You saw, like, for GWAS, you need tens of thousands or even hundreds of thousands of people to find those effects. But then with the BioResource, we can precisely match the people that we want to study. So that helps a lot. And we get access to fresh samples and also other phenotypes that you will hear more about in the other studies. And it's a very cost-effective way we can do very deep analysis, like omics, metabolomics, proteomics. That is still very expensive, but that gives us all these answers and networks and really big insights into the biology. And through this sort of studies, we can uncover novel pathways that are involved in intermediate traits such as blood cell traits and also cardiovascular disease risks. I'm going to hand over back to Amy for an overview of the other studies that we're doing.
Thank you, Lisa. So now I'll tell you a bit about the CAD Bio study. So this study investigates the role of 3 gene variants that affect cardiovascular disease risk. And here I'll present 2 examples. The first one is the PNP-LA3 substudy, which focuses on the PNP-LA3 gene. And this is thought to play a role in the development of fatty liver disease and coronary disease. And we study exactly how this variant affects the processing of fats in response to a dietary challenge. Volunteers are required to make 2 study visits on consecutive days. In the afternoon of day 1, they are asked to complete a medical questionnaire and have their height, weight and blood pressure measured. They donate a blood sample and are then provided with an energy balanced dinner, such as a jack potato with chili and cheese. They can return home overnight, but must not eat for 8 hours prior to visit 2. Early on day 2, volunteers donate a fasted blood sample and are then provided with a high carbohydrate dietary challenge meal for their breakfast. This can be cereal and toast or porridge with a glass of orange juice. A final blood sample is donated following the meal. The blood sampling at these various time points enables us to look at detailed lipid profiling, especially triglyceride levels in the blood, and compare this both before and after the high carbohydrate challenge. This study is ongoing, but we hope it may provide mechanistic insight into how to therapeutically target, manage and reduce the levels of secreted lipids from the liver into the bloodstream and to reduce the risk of coronary heart disease. The EDNRA substudy investigates the role of the EDNRA gene in coronary artery disease by measuring changes in the contraction of arteries in volunteers. As well as the medical questionnaire, height, weight, blood pressure measurements, and the blood sample, we will measure participants' forearm blood flow over 2 hours. Here, the volunteer will lie flat with their hands and arms raised above their heart, supported with pillows and foam pads, as you can see in the image here. Wrist and upper arm cuffs will inflate and deflate multiple times throughout the procedure. Fine wire loops placed around the middle of the forearm allow the volume of the arm to be measured during the cycle of inflation and deflation of the cuffs. This procedure will also include an infusion of an active substance in a very small concentration which only acts locally on the arm and not around the rest of the body. We will measure the volume of blood being pumped by the heart before and throughout this infusion period, and this method measures the volume by looking at how well or low electrical current is sent around the body. This study is ongoing, but our prediction here is that the genetic variant increases the amount of smooth muscle cells, thereby increasing arterial contractility and thus leading to an increased risk of coronary artery disease and large artery stroke. Data obtained from this study could suggest a therapeutic target for coronary heart disease or stroke prevention. Finally, to CAD Bio. This is quite an exciting study we have planned. We are looking at the risk for aortic aneurysm, which is a serious if rare occurrence. AAD stands for aortic aneurysm and dissection. You can see in the diagram a healthy artery at the top, and next a breach in the inner wall of the artery, allowing blood flow between the inner and outer walls. If the flow increases, breaking through the outer wall, as seen on the far left, blood can escape. For the CAD Bio study, we will compare results from carriers and non-carriers of particular genetic variations. Healthy participants who have agreed to take part will have their arteries scanned for baseline information. We'll take a blood sample, again for baseline measurements, and we will use stem cell technology to develop smooth muscle cells, as seen in blood vessel walls. We will follow the health of volunteers for some years and study whether these genetic variations have an effect on future health. So like many other projects, this is a long-term study. Patients with genetic variations have been shown to be at increased risk for developing aneurysm and could be monitored with a view to preventing an emergency. The BioResource have several studies recruiting for special projects in rare diseases and have added aortic aneurysm to the portfolio as this is a rare disease. This will additionally contribute genetic information from AAD sufferers and their relatives. So what impact does all of this have for patients and the public? Over the years, the techniques and resources available for research are increasingly sophisticated and enable us to take an in-depth look at genetic variations and their metabolic consequences. The NIHR BioResource is invaluable. These recall studies using human volunteers are hugely important. They give insights into disease mechanisms, and the results are directly translatable into potential treatments. The translation of results is what will help individuals suffering from these cardiovascular diseases, and this is our ultimate goal. We have seen recall studies make a real difference to participants. In this example here, individuals with a non-functional APOC3 gene have lower plasma triglycerides, an observable blunting of the usual rise in triglycerides after a meal. So you can see that in the graph. The APOC3 inhibitors are currently in clinical development, and these will reduce the production of triglyceride-rich lipoproteins with their unwanted side effects in the cardiovascular system. And this is all as a result of a Recoil by genotype study. So to summarize, this is a continual process, adding to scientific and medical knowledge, and using this to adapt, refine, and develop treatments. Each step adds to the knowledge base and is disseminated to the community through the publication of results in scientific journals, on departmental websites, in presentations, and increasingly via social media. We would like to thank all our supporters and especially the volunteers who gave their time and samples for these and in fact all research studies. And we noted just some of our funders there on the slide as well. If you are interested in helping, although we are looking at particular genes in our studies, you can still help by signing up to the BioResource at one of their centres around the country. It is entirely voluntary, and you can participate in any study to which you're invited. When invited, you can choose whether to take part, and you can withdraw if you wish at any time. If you would like to revisit this presentation at any time, it should be uploaded to the Cardiovascular Epidemiology Unit YouTube channel very soon. And also to note on that slide is the Cardiovascular BioResource studies. This is a website we've set up as part of these recall studies, and that will give you a lot more information, so you can go and view that there. So that concludes the end of our presentation section, and we would now like to open the floor to the audience to ask if you have any questions. So please either type your question in the chat box or use the raise your hand function. I can read out your questions, or if you wish, you can unmute yourself to answer your question, to ask your question.
So there's a question in the chat from Irene. Can you tell if each specific genetic difference correlates to developing into disease when certain environmental or lifestyle choices are made? So that's an interesting question. I guess there's two parts to the answer. We would refer to this as a gene-environment interaction. So that is to say, just what you've said, a genetic variant interacting with some lifestyle factor, smoking, diets, lifestyle, and that either ameliorates the risk caused by the variant or increases it. So there are examples of that, but typically these are quite difficult to demonstrate from a statistical point of view, because the sample size required is enormous to look at tens or hundreds of thousands of genetic variants along with environmental risk factors simultaneously. So typically that's been difficult. However, one of the key questions, I guess, for clinicians is if patients are, or not patients, or if individuals are receiving genetic tests, what impact does that have upon their lifestyle? So one option would be it would cause fatalism. So if you're told you've got a genetic variant that increases your risk of heart disease, then that may actually have a negative impact upon your health, and you think, well, I've got this variant, there's nothing I can do about it. You know, and it actually is negative in their health. But there are reasonably good data that demonstrate that individuals with high genetic risk can ameliorate their risk using standard lifestyle measures. And so I think that's really important, that there are data that even if you carry a variant that, or a number of variants that increase your risk of having heart disease, you can reduce the risk with fairly standard measures. And a key question for us as a community is how we routinely introduce that to healthcare. And I guess that's where the next 10, 15 years where our applied genomic research in healthcare is really going to become very important.
Thank you, Seamus. There's another question that's come in, probably also directed to Dirk and yourself. How much of a problem does unrepresentative patient samples pose in the research?
So I can take that question, maybe. So thanks very much for the question. It's a very important one. And in fact, it's a question that I think the community struggles quite a lot in terms of genetic analysis of underrepresented population. So our studies use the NIHR BioResource, and that's a normal representation of the UK. And as it happens, there are only a small proportion, so 5 to 10% of the BioResource that are from non-European ancestries. So these are therefore not represented in the cohort. So we can't necessarily recall these individuals and have a representative assessment across the different ethnic backgrounds. So that's not just a difficulty for our studies. I think in general, the community has recognized that non-European ancestry genetic data sets are lacking or lag behind. And I believe in the next few years, there will be many more genetic data sets being created that relate to non-European ancestry individuals. And hopefully, you know, that will address some of the disparity here. And hopefully our studies will then be able to translate to different backgrounds and hopefully inform the more general population.
Thank you, Dirk. I have another question here related to the COVID situation we're in at the moment. So how has COVID-19 affected recruitment to your studies? Dirk, would you like to take that one, or Seamus?
Yeah, but badly, really. The BioResource was impacted upon significantly by the pandemic. So it is back and running. It took a little while, but I think that's probably the short, sharp answer. But badly.
That's right. I think a lot of the resources and capacity at the BioResource, or the nurses who work there, have been reallocated to projects relating to COVID, and rightfully so. So all studies that involved healthy volunteers and that addressed questions unrelated to COVID were paused and are about to restart now that the pandemic is hopefully easing.
Oh, another question that's just come in. Are there any new technologies such as genetic engineering which could aid in reducing such diseases?
I can take that question. So that's, you know, that's super important and very interesting. In fact, genetic engineering using so-called CRISPRs, which you may have come across, was actually used as part of Lisa's PhD project. So while genetic engineering methods are not readily applied in humans, it's certainly a technique that we have applied to validate some of our findings in humans that related to just puncture and assessment of blood cells from that material. So that's certainly something we can do in the lab. It's not something that is easily to be translated into humans. However, there are some efforts ongoing that use such technology for curing heart disease. I think one of the examples that were given at the end by Amy, where studies are in clinical trials, some of them use advanced technology, for example, antisense oligonucleotides that target particular genes of interest and knock it out, so reduce the levels of these genes to ameliorate the disease. So I think more and more new technologies and modalities are being used, both in academia for basic research, but also for clinical trials.
Thank you, Dirk. I have another question here. How does the BioResource know which studies to support?
Well, I guess there's two answers to that. One is which diseases do they support, and initially they supported a panel of rare diseases, and the reasons for that are rare disease research is challenged in an individual center, because obviously in any one place there aren't that many patients with a rare disease, so having a national infrastructure was a way to tackle that. They will also move on to more common diseases, and really which ones they support will be determined by the scientific needs expressed by the researchers interested in that area. How do they support individual studies on individuals rather than, healthy individuals rather than those with disease? Each study that, like the one that Lisa reported, has its own application and its own funding process, which is reviewed centrally by the BioResource in a pretty timely fashion, really. And as with everything, the key thing is what is the scientific question being asked, and what is the safety and so on of the approach being proposed.
That's right. And just to add, so all the studies require ethical approval. So even the scientific rationale might be strong. It is also super important that, of course, any methods or technologies that we use and are tested on these healthy volunteers really comply with the ethical boundaries. So everything is very thoroughly checked. And it's also important to note that for the BioResource in particular, individuals are only contacted up to 4 times a year. So it may be that there's a need to recall particular individuals more often, but this will not be allowed by the BioResource. So 4 times maximum for individual studies.
Thank you, Dirk. I've got a couple more questions. Oh, no, there's 3 now. So I'll just read them all out and then maybe we can see which ones we can answer. Is there a gender difference in the incidence and number of genetic variants? How do you think your research will impact insurance companies in the future?
So I'll take perhaps the second question first. I think we don't know is the answer, and it's clearly a concern for the community and for individuals. In the UK, it's probably less at the forefront of our mind than in other places because of the healthcare system we have here. However, I think what the question's alluding to is if you have genetic sequencing as a young person and identifies a particular risk, would that negatively impact upon your ability to be insured? I guess the way to approach that is going to be centralized guidelines and consideration, and not introducing the technology too soon, because frankly at the minute we don't quite know what a lot of the variants do, and I don't think our knowledge is strong enough to be impacting on things like that. But it's a really tricky ethical area. In terms of the gender difference, so before we spoke about gene environment interactions, and I suppose that's a form of again, interaction. Do the variants have different effects on males and females? In some cases, yes, and obviously there are genetic variants on the specific sex chromosomes, which will be different between men and women. But in general, most of the variants that we find have a similar effect on men and women. Dirk, did you want to add anything to either of those?
No, I think you summarized it well. And maybe Amy, I can take the final question on epigenetics. So the question is, how do you see epigenetics in this context, and any plans to consider it in your studies? So as part of our studies that we presented today, we haven't considered epigenetics in particular, but clearly that's a very exciting area of work, it certainly applied previously. So there's, for example, studies that have been done as part of the BioResource, recruiting 200 healthy individuals for a huge European-led project called Blueprint, and that aimed to assess the epigenetic profiles of human blood cells and to correlate these findings with disease. So that's certainly something that can be done in this context and has been done before.
I've had one more question directly sent to me relating to the example that was shown at the end there, the APOC3 variant. It seemed to affect both the mean levels and variability with variance, especially increasing in the control group. How does that limit inferences in the context of the small sample sizes that they were limited to studying?
Yeah, that's a very tricky question. As has already been commented on, I think for this particular study, the sample size was very small, and therefore the error bars of this observation are rather large. I think while it's true that it appears to also affect variability, that could just reflect differences between the studied individuals, say, BMI, a different age, where you might expect differences that are somewhat unrelated to the study. So just because you have smaller sample sizes than potentially in larger population-based studies. Though is pretty remarkable that even in the small sample size and the large error bars, it is a very clear result. So you can really see that the cases have a markedly different response to oral fat challenge. And indeed, in subsequent clinical trials that now involve much larger populations, you can really see a marked difference on triglyceride levels when you inhibit ApoC3. So I think what is important to note that these recall studies based on small sample size are essentially just an avenue towards understanding mechanism. It is not a definitive answer of understanding biology, and I think all the studies require independent validation and larger data sets, and then of course randomized clinical trials to really ascertain an effect.
Okay, I think we've got time for one last question. You have mentioned you use healthy individuals in your trials, but do you do any research with already diseased subjects? So I think this might be for you, Seamus.
I mean, I think the quick answer is yes. We do both healthy individuals and individuals with disease. Using healthy individuals is relatively new. The standard approach is to compare healthy individuals to individuals who carry a particular disease.
I think it depends really on the research question. So Seamus is absolutely right. So depending on what kinds of mechanisms you'd like to understand, you may choose healthy individuals, which has an advantage of not being perturbed by the actual disease or by the medical treatment, for example. So here, those studies are particularly useful for studying basic biological processes, like Lisa mentioned. But then if you really want to have a more disease context, like Seamus's CAD Bio study will be able to address, then it is a requirement to really look for individuals with the disease.
Okay, thank you very much for all your questions, and thank you to the panel for answering them. Finally, we have a second poll, which we would like to know what you now know about cardiovascular disease. Has this presentation helped in any way to give you more information and knowledge about the topic? So you should be able to see the question on the screen now, if you wouldn't mind answering what you now feel you know or understand about the subject of genetics and cardiovascular disease. I'm also going to drop a link in the chat box, which is to an audience snapshot survey. So on behalf of the Cambridge Festival, they would like to get some feedback from you. It's a short 5-question survey, so it should just take a minute. And the more people that complete it, the more reliable their data is, and to help them plan and prepare events in the future. So that link's in the chat box for you. Did anyone else want to give their opinion in the voting? There's about 75% people that have voted. But the majority say that they've considerably improved their knowledge about cardiovascular disease, which is very encouraging following our presentation. Okay, so I think we'll finish the voting there. Thank you very much to everyone that's voted. And thank you for everyone for attending. I hope you found the presentation and information useful, and a valuable discussion with our panel members. Thank you, Dirk and Seamus. And thank you to Lisa for sharing your results from your PhD as well. Okay, thank you, everybody.